Hello, dear student. So, during the last lecture, we have seen what is a fat-free or skimmed milk, organic milk, and lactose-free milk. So, this is what the types of, these are the three types of milks. And now we are going to see the analysis of milk for lactose by Lane-Anon method. Lane-Anon method. So main carbohydrate in the milk is the lactose, that is about 50%, and it gives milk a sweet taste. Trace amount of other sugar like glucose, fructose are also present. So lactose is a disaccharide and consists of glucose and galactose moieties linked through 1,4 glycosidic bond. Glycosidic bond. Okay, it is some problem is there. Then the hemiacetal group of a glucose moiety is a potentially free. Hence, glucose is a reducing sugar capable of reducing certain oxidizing agent. Okay, now see. This method is based on the reducing property of lactose from the milk. Reducing sugar contains free aldehyde groups, and that can reduce the metals like copper, iron, silver under the alkaline medium. This property is used in the failing solution. Okay, which is a mixture of failing solution A, that is a copper sulfate pentahydrate, and failing solution B, alkaline sodium potassium tartarate. When the solutions are mixed, copper sulfate in the alkaline medium is precipitated as a copper hydroxide. However, presence of sodium potassium tartarate, it forms a soluble complex which behaves like alkaline copper, behaves as alkaline copper hydroxide solution. Alkaline copper hydroxide solution. When Felix solution is heated, it forms a cupric oxide. So when we are heating that solution, it forms a cupric oxide, and which in turn, with the reducing sugar, get reduced to form a cuprous oxide. Okay, this is cupric oxide, and this is a cupric. Okay, it gives big red precipitate. Sugar get oxidized to corresponding acid. Gradual color changes make it difficult to detect the end point. Hence, Lane-Anon in 1923 introduced methylene blue to this process as an indicator for detection of endpoint when titrating a failing solution with the reducing sugar. Okay, so what actually the reagents are there? See, a failing solution A, that is copper sulfate pentahydrate. Failing solution B, where KOH and sodium potassium tartarate are mixed together in the solution B. Then 5 normal HCl, 1% methylene blue is there, 1% lactose solution is there, and 10% acetic acid is there. Okay, so you have to just remember the solutions. Okay, and there is no need to remember how that can be prepared. That is available in the literature, but you must know we are using failing solution A, B, 5 normal HCl, 1% methylene blue, 1% lactose solution, 10% acetic acid solution. Now, sample solution. How we are going to prepare it? Take 1 gram of milk powder and transfer to 50 ml of beaker. Add 15 ml of distilled water and warm to dissolve the powder. The precipitate the protein by adding 1 or 2 drops of acetic acid. त्यात acetic acid टाकलं की protein काय होणार आहे त्याच्यामध्ये precipitate होतील. Digest the precipitate on water bath for half an hour. Filter the solution through Wattmann filter paper number 42 and collect the filtrate in 250 ml of standard flask and dilute it up to the mark. Okay. 250 ml ला ते dilute करायचं आहे जे आपलं filtrate आलेलं आहे ते. Now standardization of failing solution. take 5 ml of filling solution A, 5 ml of filling solution B in a dry evaporating dish. Boil the content by heating on a wire gauze. Then titrate the above boiling solution against 1% lactose solution till the blue color discharge disappears. To this solution, add after disappearance of blue color, add 3 to 4 drops of methylene blue indicator. Continue the titration till the color of indicator discharge. Okay, methylene blue. That color of methylene blue should be get disappear. At the end point, the blue color of the solution should be discharged, and supernatant should be yellow with a brick red precipitate of cuprous oxide. Okay, that should be the cuprous, right? This one, right? Cuprous. So note down this as a reading V1 ml. Note down this as a V1 ml. After that, now pipette out 5 ml of filling solution A, 5 ml of filling solution B in a evaporating dish. Boil the content on a wire gauze. Then to this add a the titrate the above boiling solution against the sample solution till the blue color get discharged. To this add 2 to 3 drops or 3 to 4 drops methylene blue indicator. Continue the titration till the blue color get discharged. At the end point, blue color of the solution get discharged. The supernatant should be yellow with brick red precipitate. Okay, so brick red precipitate. So and note down this as a reading V2. So here it was a reading standardization of filling solution. It was a reading V1, and estimation it was a reading V2. The percentage of lactose in the milk is 5 into V1 upon V2. After that, a new point is there, adulterant in the food products. Adulterants in the food products. Okay, so we'll continue that part in the next lecture. There are different adulterant people used to add in it, and that adulterants are having very adverse effect on human body. And that the addition of adulterant is for the economical benefit of the people who are delivering the milk. That is very bad practice in actually day-to-day life. And only chemist can analyze what adulterants are there, whether adulterants are added in the milk or not. So that we are going to continue in the next lecture. Thank you.